Welcome to Sleepy Science Odyssey. Tonight, DNA, the molecule that stores the instructions for life, from the double helix discovered in 1953 to the 3 billion base pairs encoding human biology, from the mechanisms of replication ensuring faithful copying to the mutations creating variation, from genes coding for proteins to regulatory sequences controlling expression, from inheritance patterns passing traits through generations to genetic engineering allowing precise modifications. We'll explore what DNA is, how it works, how it's passed on, how it changes, and why understanding this molecule matters for understanding life itself. If you're new here, this channel is for people who need something to think about before they can stop thinking. Real molecular biology and genetics, no exaggeration, just quiet facts in the dark. Let's begin. DNA is a double helix composed of 2 complementary strands of nucleotides running in opposite directions. The structure of DNA represents one of the most important discoveries in biology, revealing how genetic information can be stored, replicated, and transmitted with extraordinary fidelity across generations. Understanding DNA structure provides the foundation for understanding all of molecular biology, from how cells function to how organisms develop, from how evolution operates to how diseases arise. DNA stands for deoxyribonucleic acid, a long polymer molecule composed of repeating units called nucleotides. Each nucleotide consists of 3 components: a sugar molecule called deoxyribose, a phosphate group, and one of 4 nitrogenous bases: adenine, thymine, guanine, or cytosine, commonly abbreviated A, T, G, and C. The sugar and phosphate groups form the backbone of DNA strands, with the bases projecting inward where they can interact with bases on the complementary strand. The double helix structure was determined by James Watson and Francis Crick in 1953, based on X-ray crystallography data collected by Rosalind Franklin and Morris Wilkins. The structure revealed that DNA consists of 2 strands wound around each other in a right-handed helix, resembling a twisted ladder where the sugar-phosphate backbones form the sides and paired bases form the rungs. This structure immediately suggested how DNA could be copied. Each strand could serve as a template for creating a new complementary strand. The 2 strands of the double helix run in opposite directions, termed antiparallel. DNA strands have directionality based on the asymmetry of the sugar-phosphate backbone, with one end called 5' and the other end called 3' based on the carbon numbering in deoxyribose. In the double helix, one strand runs 5' to 3' in one direction, while the complementary strand runs 5' to 3' in the opposite direction. This antiparallel arrangement is crucial for DNA replication and has important consequences for how DNA is copied. Base pairing follows strict rules that were immediately apparent from the structure. Adenine always pairs with thymine through 2 hydrogen bonds, while guanine always pairs with cytosine through 3 hydrogen bonds. These Watson and Crick base pairs have complementary shapes and hydrogen bonding patterns that allow them to fit together precisely within the double helix while excluding other combinations. The AT and GC pairing means that if you know the sequence of one strand, you can deduce the sequence of the complementary strand. They contain the same information in complementary form. The width of the double helix is remarkably uniform at approximately 2 nanometers because purine-pyrimidine base pairs, AT and GC, have very similar dimensions. Purines, adenine, and guanine are larger molecules with 2 fused rings, while pyrimidines, thymine, and cytosine are smaller with single rings. Each Watson and Crick pair consists of one purine and one pyrimidine, maintaining constant width. If two purines paired, the helix would bulge. If two pyrimidines paired, it would narrow. The strict pairing maintains structural regularity. The helical structure repeats every 10 base pairs, completing one full turn of the helix approximately every 3.4 nanometers along the long axis. This regular helical geometry creates major and minor grooves spiraling along the outside of the helix, where proteins can access the edges of base pairs without disrupting base pairing. Many DNA-binding proteins recognize specific sequences by reading the pattern of hydrogen bond donors and acceptors, and hydrophobic and hydrophilic groups in the major groove, allowing sequence-specific protein-DNA interactions crucial for gene regulation. The stability of the double helix derives from multiple interactions. Base pairing through hydrogen bonds contributes to stability, but equally important are base stacking interactions. The van der Waals forces between the flat aromatic rings of adjacent bases stacked like plates along the helical axis. These stacking interactions provide substantial stabilization and explain why DNA stability increases with length. Longer DNA molecules have more base stacking interactions. The hydrophobic effect also contributes, with the relatively hydrophobic bases buried in the helix interior away from water, while the hydrophilic sugar phosphate backbone faces outward. DNA can adopt different conformations depending on conditions. The standard B-form DNA described by Watson and Crick occurs under physiological conditions with moderate humidity and salt concentrations. A form DNA occurs under low humidity or high salt conditions, with a wider, more compact helix and base pairs tilted relative to the helical axis. Z-form DNA occurs in specific sequences, particularly alternating GC sequences, forming a left-handed helix. These alternative forms may have biological functions, with zDNA possibly involved in gene regulation, though B-form predominates under physiological conditions. The length of DNA molecules varies enormously. The human genome contains approximately 3 billion base pairs distributed across 23 pairs of chromosomes. The longest human chromosome, chromosome 1, contains approximately 250 million base pairs, which if stretched out would extend approximately 8 centimeters, remarkably long for a molecule, yet microscopic in width. The entire human genome, if all chromosomes were joined end-to-end, would extend approximately 2 meters. Fitting this into a nucleus approximately 10 micrometers in diameter requires extraordinary compaction. DNA compaction involves multiple levels of organization. DNA wraps around histone proteins to form nucleosomes, the basic unit of chromatin, with approximately 150 base pairs wound around each histone octamer. Nucleosomes connected by linker DNA resemble beads on a string. This nucleosome string coils into 30 nanometer chromatin fibers, which form loops anchored to protein scaffold, which condense further during cell division to form the familiar X-shaped chromosomes visible under microscopes. This hierarchical compaction achieves approximately 10,000-fold reduction in length. The chemical stability of DNA under physiological conditions is remarkable but not absolute. DNA is vulnerable to damage from multiple sources, including spontaneous hydrolysis breaking chemical bonds, oxidative damage from reactive oxygen species, UV radiation causing thymine dimers, and various chemical modifications. Cells possess elaborate DNA repair systems that detect and correct most damage, maintaining genome integrity. However, unrepaired damage can cause mutations, permanent changes in DNA sequence that may be harmful, neutral, or occasionally beneficial. The negative charge of DNA due to phosphate groups has important consequences. DNA is highly negatively charged, with each phosphate contributing one negative charge, making DNA polyanionic. This negative charge causes DNA to strongly interact with positively charged molecules, including histone proteins, which have many positively charged amino acids that neutralize DNA's negative charges, allowing tight packing. The negative charge also makes DNA soluble in water and causes electrostatic repulsion between DNA molecules, preventing them from spontaneously aggregating. DNA's chemical composition distinguishes it from RNA, the related nucleic acid. DNA contains deoxyribose sugar, while RNA contains ribose, which has an additional hydroxyl group at the 2' carbon position. This seemingly minor difference has major consequences. The 2' hydroxyl in RNA makes it chemically reactive and prone to hydrolysis, while DNA lacking this group is more stable, making DNA better suited for long-term information storage. DNA also uses thymine, while RNA uses uracil, another difference whose significance relates to damage detection and repair. The amount of DNA in cells varies across organisms, but not always in ways that correlate with complexity. Humans have approximately 6 picograms of DNA per diploid cell, about 3 billion base pairs in each copy of the genome. The smallest bacterial genomes contain fewer than 200,000 base pairs, while some single-celled amoebas have genomes over 100 times larger than humans. This C-value paradox, the lack of correlation between genome size and organism complexity, reflects varying amounts of non-coding DNA rather than differences in gene number. DNA replication creates 2 identical copies from one double helix through semi-conservative replication. The mechanism by which DNA copies itself was predicted by the Watson and Crick structure, which immediately suggested that the 2 strands could separate and each serve as a template for synthesizing a new complementary strand. The experimental demonstration of this semi-conservative replication mechanism and the elucidation of the molecular machinery carrying it out represent major achievements of molecular biology. Semi-conservative replication means that each of the 2 daughter DNA molecules contains one original strand and one newly synthesized strand. This contrasts with conservative replication, where one daughter would contain both original strands and the other would contain 2 new strands, or dispersive replication, where each daughter would contain segments of old and new DNA intermixed along each strand. The semi-conservative model was proven by the Michelson-Stahl experiment in 1958, one of the most elegant experiments in biology. Michelson and Stahl grew bacteria in medium containing heavy nitrogen-15, which incorporated into DNA, making it denser than normal DNA containing nitrogen-14. After many generations, all DNA was heavy. They then transferred bacteria to normal nitrogen-14 medium and allowed one round of replication. The resulting DNA had intermediate density, consistent with each molecule containing one heavy strand and one light strand, as semi-conservative replication predicts. After a second round of replication, half the DNA was intermediate density and half was light density, exactly as expected. DNA replication begins at specific sites called origins of replication, where the double helix is open to allow access to the template strands. In bacteria, which have circular chromosomes, replication typically begins at a single origin and proceeds bidirectionally around the circle. In eukaryotes, which have much larger linear chromosomes, replication begins at thousands of origins distributed along each chromosome, with replication proceeding bidirectionally from each origin until adjacent replication forks meet and fuse. The opening of the double helix creates replication forks, Y-shaped structures where the two strands have separated. Multiple proteins work together to initiate and stabilize replication forks. Helicase enzymes use energy from ATP hydrolysis to break the hydrogen bonds between base pairs and unwind the helix. Single-strand DNA-binding proteins, or SSBs, coat the separated strands, preventing them from reannealing and protecting them from nucleases. Topoisomerase enzymes relieve the tension created ahead of replication forks by temporarily breaking and rejoining DNA strands, preventing the accumulation of positive supercoils that would halt replication. DNA polymerases, the enzymes that synthesize new DNA strands, have a fundamental limitation. They can only add nucleotides to the 3' end of existing DNA or RNA strands, meaning they require a primer to initiate synthesis. This requirement is met by primase, an RNA polymerase that synthesizes short RNA primers approximately 10 nucleotides long complementary to the DNA template. DNA polymerase then extends these RNA primers, adding deoxyribonucleotides to create new DNA strands. The antiparallel nature of DNA strands combined with DNA polymerase's 5' to 3' synthesis direction creates a problem. At each replication fork, one template strand runs 3' to 5' in the direction of fork movement, allowing continuous synthesis of the new strand in the same direction as fork movement. This is the leading strand. The other template strand runs 5' to 3' in the direction of fork movement, requiring that the new strand be synthesized in the opposite direction from fork movement. This is the lagging strand. Leading strand synthesis is relatively straightforward. One RNA primer is synthesized at the origin, and DNA polymerase continuously synthesizes new DNA in the 5' to 3' direction following the replication fork. Lagging strand synthesis is more complex. As the fork opens, primase repeatedly synthesizes new RNA primers, and DNA polymerase synthesizes short segments called Okazaki fragments, each approximately 1,000 to 2,000 nucleotides in bacteria or 100 to 200 in eukaryotes, in the direction opposite to fork movement. The Okazaki fragments must be joined to create a continuous lagging strand. This maturation process involves multiple steps. RNase H and Fen1 nuclease remove RNA primers. DNA polymerase fills the gaps left by primer removal. DNA ligase joins the fragments by forming phosphodiester bonds between the 3' hydroxyl of one fragment and the 5' phosphate of the next. The result is a continuous DNA strand identical to leading strand products despite the complex synthesis process. DNA polymerases have extraordinary fidelity, making errors at rates of approximately 1 mistake per 100,000 to 1 million nucleotides incorporated. This accuracy derives from multiple mechanisms. The geometry of the polymerase active site strongly favors correct Watson and Crick base pairs over mismatches. Induced fit conformational changes occur when correct nucleotides bind, positioning catalytic residues optimally. Most importantly, DNA polymerases possess 3' to 5' exonuclease activity, allowing them to remove incorrectly incorporated nucleotides immediately after addition, a proofreading function that reduces error rates approximately 100-fold. Replication is extremely rapid despite its complexity and accuracy. In bacteria, replication forks move at approximately 1,000 nucleotides per second. In eukaryotes, forks move more slowly at approximately 50 nucleotides per second, but the presence of thousands of origins operating simultaneously allows complete replication of large genomes in several hours. The coordination of initiation at multiple origins and the regulation ensuring each region is replicated exactly once per cell cycle involve sophisticated control mechanisms. Telomeres, the ends of linear chromosomes, present a special replication problem called the endreplication problem. Because RNA primers are required to initiate synthesis and are later removed, the extreme 5' ends of lagging strands cannot be completely replicated, leading to progressive shortening with each replication cycle. This shortening is counteracted by telomerase, a specialized reverse transcriptase that extends telomeres using an RNA template carried within the enzyme. Telomerase is active in germ cells and stem cells, but not in most somatic cells, contributing to cellular aging. The replication machinery forms a replisome, a large protein complex containing multiple copies of polymerases, primase, helicase, and accessory proteins. The replisome is highly processive, remaining associated with DNA through synthesis of thousands or millions of nucleotides. Processivity factors including pCNA in eukaryotes form sliding clamps that encircle DNA and tether polymerases, preventing dissociation while allowing sliding along DNA. The replisome architecture coordinates leading and lagging strand synthesis despite their different mechanisms. Replication fidelity is further enhanced by post-replication mismatch repair systems that scan newly synthesized DNA for errors missed by polymerase proofreading. These systems recognize distortions in the double helix caused by mismatched base pairs, excise the incorrect nucleotide along with surrounding sequence, and resynthesize the correct sequence. Mismatch repair reduces error rates approximately another 100 to 1,000-fold, achieving overall fidelity of approximately 1 error per billion nucleotides incorporated, a remarkable achievement given the speed of replication. DNA replication is regulated to ensure it occurs exactly once per cell cycle in the S phase, separating the two gap phases. This regulation involves multiple mechanisms, including licensing of origins during G1 phase, when replication proteins assemble at origins, but synthesis doesn't begin. Initiation of synthesis at licensed origins during S phase, and mechanisms preventing relicensing during S, G2, and M phases. Dysregulation of replication control contributes to genomic instability and cancer. The energetic cost of DNA replication is substantial. Each nucleotide incorporated requires hydrolysis of nucleoside triphosphates, the building blocks of DNA, which provide both the nucleotide and energy for synthesis. Additional ATP is consumed by helicases unwinding DNA, by primase synthesizing primers, and by other replication proteins. Despite these costs, faithful genome duplication is essential for inheritance and organismal survival, justifying the investment. Genes are segments of DNA that encode instructions for making proteins or functional RNAs. The concept of genes as units of heredity predates the discovery of DNA structure, but the molecular understanding of genes, what they are, how they work, how they're organized, emerged from molecular biology. Genes represent the functional units of genomes, the sequences that contain information for making the molecules that carry out cellular functions. A gene in its simplest form consists of a DNA sequence that is transcribed into RNA. For protein-coding genes, the RNA is messenger RNA or mRNA, which is translated into protein. For RNA genes, the RNA itself is the functional product, including ribosomal RNAs that form ribosomes, transfer RNAs that carry amino acids during translation, and various regulatory RNAs. The distinction between genes and non-coding DNA was historically clear, but modern genomics has blurred boundaries by revealing extensive transcription of sequences previously considered non-functional. Protein-coding genes have characteristic structures in eukaryotes. They begin with promoter regions, where transcription machinery binds to initiate RNA synthesis. The transcribed region includes exons, sequences that appear in mature mRNA and often introns, intervening sequences that are transcribed but removed during RNA processing. The coding sequence begins with a start codon, typically ATG, and ends with a stop codon, TAA, TAG, or TGA. Untranslated regions, or UTRs, flank the coding sequence, appearing in mRNA but not being translated into protein. The human genome contains approximately 20,000 to 25,000 protein-coding genes, fewer than initially predicted before genome sequencing. This number is only modestly larger than simpler organisms. The roundworm C. elegans has approximately 20,000 genes, and the fruit fly Drosophila has approximately 14,000, highlighting that organismal complexity doesn't correlate simply with gene number. Humans achieve complexity through alternative splicing, where single genes produce multiple protein variants, and through complex gene regulation. Gene size varies enormously. The smallest human genes span fewer than 1,000 base pairs, while the largest, dystrophin, extends over 2 million base pairs. The size difference reflects primarily the presence of introns. Dystrophin contains 79 exons interrupted by 78 introns, some exceeding 100,000 base pairs. The exons total only approximately 14,000 base pairs, coding for a protein of approximately 3,700 amino acids, but the introns vastly expand the gene size. Gene density, the number of genes per unit length of DNA, varies across genomes and across chromosomes. In compact bacterial genomes, genes occupy 80 to 90% of sequences with minimal non-coding DNA. In the human genome, genes occupy only approximately 1.5% of sequences, with vast intergenic regions containing repetitive sequences, transposable elements, and sequences of unknown function. Some chromosomal regions are gene-rich while others are gene-poor, creating heterogeneity in gene distribution. The discovery that genes contain introns, sequences that are transcribed but spliced out before translation, was surprising and transformed understanding of gene structure. In 1977, Philip Sharp and Richard Roberts independently discovered introns in adenovirus genes, overturning the assumption that genes were continuous coding sequences. Introns are common in eukaryotic genes, but rare in bacteria, representing a major difference between prokaryotic and eukaryotic genome organization. Alternative splicing of introns allows single genes to produce multiple mRNA and protein variants. Exons can be included or excluded, intron-exon boundaries can vary, and different combinations can be used in different cell types or conditions. Approximately 95% of human multi-exon genes undergo alternative splicing, vastly expanding the proteomic diversity encoded by approximately 22,000 genes. Some genes produce dozens or hundreds of variants through combinatorial splicing. Gene regulation determines when, where, and how much gene expression occurs. Regulatory sequences, including promoters, enhancers, and silencers, control transcription initiation. Transcription factors, proteins that bind specific DNA sequences, activate or repress transcription depending on cellular conditions, developmental stage, and environmental signals. The combinatorial action of multiple transcription factors allows precise spatio-temporal control of gene expression, enabling development, differentiation, and responses to environment. Enhancers are regulatory sequences that can be located far from the genes they regulate, sometimes hundreds of thousands of base pairs away, and can act independently of orientation. Enhancers contain binding sites for multiple transcription factors, and when appropriate combinations of factors bind, the enhancer loops through three-dimensional space to contact the promoter, stimulating transcription. This looping mechanism allows flexible long-range regulation and explains how mutations far from genes can affect their expression. Epigenetic modifications, chemical modifications of DNA or histones that affect gene expression without changing DNA sequence, provide additional regulatory layers. DNA methylation, the addition of methyl groups to cytosine bases, particularly in CG dinucleotides, generally represses transcription. Histone modifications, including acetylation, methylation, phosphorylation, and ubiquitination, affect chromatin structure and gene accessibility. These modifications can be maintained through cell divisions, providing cellular memory of gene expression states. Gene families arise through gene duplication followed by divergence. Duplication can occur through unequal crossing over transposon-mediated duplication or whole genome duplication. Duplicated genes may acquire mutations causing loss of function, retention of original function, providing redundancy, or gain of new function, neofunctionalization. Gene families allow specialization and evolution of related functions while maintaining original functions. The globin gene family, including myoglobin and multiple hemoglobin chains, exemplifies functional diversification following duplication. Pseudogenes are gene sequences that have lost function through disabling mutations while retaining similarity to functional genes. Processed pseudogenes arise through reverse transcription of mRNA followed by insertion into genomic DNA, producing intronless copies lacking regulatory sequences. Duplicated pseudogenes arise through duplication followed by mutation. Pseudogenes were considered genetic fossils without function, but some appear to regulate their parent genes through RNA interference or other mechanisms, challenging simple classifications. The concept of what constitutes a gene continues to evolve. Classical definitions based on coding sequences are inadequate for non-coding RNA genes. Functional definitions based on transcription include many more sequences than coding-centric definitions. Some propose that genes should be defined by function rather than sequence properties, including regulatory sequences as parts of genes. The ENCODE project's finding that approximately 80% of the human genome is transcribed has sparked debate about how much is functional versus transcriptional noise. Gene nomenclature systems provide standardized ways to name and describe genes. Human genes use symbols consisting of uppercase letters, and numbers following specific conventions, with different species using different conventions. Gene names often reflect function, homology to known genes, or historical discovery circumstances. Databases including GenBank, Ensemble, and NCBI maintain gene annotations, sequences, and functional information. Standardized nomenclature facilitates communication and data sharing across the research community. Transcription converts DNA sequences into RNA molecules through the action of RNA polymerases. The process of transcription, synthesizing RNA from DNA templates, represents the first step of gene expression, converting genetic information stored in DNA into RNA molecules that either function directly or serve as templates for protein synthesis. Understanding transcription mechanisms illuminates how cells control gene expression and respond to developmental and environmental signals. Transcription is carried out by RNA polymerases, large multi-subunit enzymes that synthesize RNA complementary to DNA template strands. Bacteria have a single RNA polymerase that transcribes all genes, while eukaryotes have three main RNA polymerases with specialized functions. RNA polymerase I transcribes most ribosomal RNAs. RNA polymerase II transcribes messenger RNAs and many non-coding RNAs. And RNA polymerase III transcribes transfer RNAs and other small RNAs. The focus here will be primarily on RNA polymerase II and mRNA transcription. Transcription initiation requires RNA polymerase to bind to promoter regions and begin RNA synthesis. In eukaryotes, RNA polymerase II cannot bind promoters directly, but requires transcription factors to recognize and bind promoter sequences. The TATA box, a sequence approximately TATA, located approximately 25 to 30 base pairs upstream of transcription start sites, is recognized by TATA binding protein, or TBP. Additional general transcription factors assemble with RNA polymerase to form a pre-initiation complex capable of initiating transcription. The transcription start site marks where RNA synthesis begins, typically designated +1. Sequences upstream toward the 5' end of the DNA template strand are designated with negative numbers, while downstream sequences toward the 3' end are positive. Promoters contain various sequence elements including the TATA box, initiator sequences around the start site, and downstream promoter elements. Not all promoters contain all elements, and some genes use alternative promoters, allowing different transcription start sites. Transcription proceeds through 3 phases: initiation, elongation, and termination. During initiation, RNA polymerase binds to promoters, locally melts the DNA helix to create a transcription bubble exposing template strand bases, and synthesizes initial RNA nucleotides. The early phase of elongation is unstable, abortive transcription, with polymerase synthesizing and releasing short RNA fragments before committing to productive elongation. Once committed, polymerase undergoes promoter clearance, escaping the promoter and transitioning to elongation. Elongation involves RNA polymerase moving along DNA, unwinding the helix ahead and reannealing it behind, while synthesizing RNA complementary to the template strand. The transcription bubble moves with the polymerase, maintaining approximately 9 base pairs of DNA-RNA hybrid and 8 to 9 bases of unwound DNA. RNA polymerase II elongates at approximately 20 to 40 nucleotides per second in vivo, much slower than in vitro rates. reflecting pausing and regulation during elongation. The nascent RNA emerges from the polymerase and can interact with RNA-binding proteins immediately. Transcription factors regulate initiation by binding enhancers or silencers and modulating polymerase recruitment and activity. Activators recruit coactivator complexes that facilitate pre-initiation complex assembly or modify chromatin to increase accessibility. Repressors recruit corepressor complexes that inhibit assembly or compact chromatin. The combinatorial action of multiple activators and repressors allows precise control, with different combinations producing different expression levels. Signal transduction pathways often target transcription factors, providing mechanisms for external signals to modulate gene expression. Chromatin structure profoundly affects transcription. DNA wrapped around histones in nucleosomes is relatively inaccessible to polymerases and transcription factors. Chromatin remodeling complexes use ATP to slide, eject, or restructure nucleosomes, creating accessibility. Histone-modifying enzymes add or remove chemical groups that affect chromatin compaction and protein recruitment. Acetylation of histone lysines generally promotes transcription by neutralizing positive charges that bind DNA, while decetylation represses transcription. Histone methylation can activate or repress depending on which lysines are modified. RNA processing occurs co-transcriptionally in eukaryotes. While transcription is still occurring, the RNA undergoes modifications. The 5' end receives a 7-methylguanosine cap shortly after transcription initiation, protecting the RNA from degradation and facilitating ribosome binding during translation. The 3' end is cleaved at a polyadenylation signal and receives a poly-A tail of approximately 200 adenines, which also protects from degradation and aids translation. These modifications convert primary transcripts into mature mRNAs. Splicing removes introns from pre-mRNA, joining exons to create mature mRNA. Splicing is carried out by the spliciosome, a large complex containing 5 small nuclear RNAs, or snRNA, and over 100 proteins. The spliciosome recognizes splice sites, conserved sequences at intron-exon boundaries, and catalyzes 2 transesterification reactions that excise introns as lariat structures and ligate exons. Splicing occurs co-transcriptionally, with early exons often spliced before transcription of downstream exons is complete. Alternative splicing produces different mRNAs from single genes through differential exon inclusion, intron retention, or alternative splice site usage. Splicing is regulated by splicing factors that bind enhancer or silencer sequences in exons or introns, promoting or inhibiting spliciosome assembly at nearby splice sites. Different cell types express different combinations of splicing factors, producing tissue-specific splicing patterns. Developmental transitions and environmental signals can alter splicing patterns, providing dynamic control of protein isoform expression. Transcription termination in eukaryotes is coupled to 3' end processing. RNA polymerase II continues transcribing past polyadenylation signals, but the transcript is cleaved at these signals, releasing the upstream fragment that becomes mature mRNA. The downstream fragment is degraded by exonucleases that eventually catch the still transcribing polymerase, causing termination. Alternatively, conformational changes after polyadenylation signal read-through may reduce polymerase processivity, promoting termination. The mechanisms remain incompletely understood. Transcriptional regulation integrates multiple signals to determine expression levels. Signal transduction pathways activate or inhibit transcription factors in response to hormones, growth factors, stress, or developmental cues. Cell cycle regulation controls expression of cyclins and other cell cycle regulators. Circadian clocks drive rhythmic expression of thousands of genes. Cellular differentiation involves massive reprogramming of transcriptional patterns. Cancer often involves dysregulation of transcription through mutations in transcription factors or chromatin regulators. The transcriptional landscape of genomes extends beyond protein-coding genes. Long non-coding RNAs or LNC-RNAs are transcribed like mRNAs, often from intergenic regions, but don't encode proteins. Some LNC-RNAs regulate nearby genes, while others have diverse functions including chromatin organization, transcriptional regulation, and RNA processing. Enhancer RNAs or eRNAs are transcribed from enhancer sequences and may facilitate enhancer function. Antisense transcripts are transcribed from the strand opposite protein-coding genes and can regulate those genes. Transcription creates opportunities for regulation at multiple steps. Recruitment of RNA polymerase to promoters is rate-limiting for many genes. Pausing of elongating polymerase allows regulation during elongation. Alternative splicing, polyadenylation, and RNA stability provide post-transcriptional control. These multiple control points create a regulatory landscape where gene expression responds precisely to cellular needs, developmental programs, and environmental conditions. Translation converts messenger RNA sequences into proteins through the action of ribosomes. Translation represents the final step of gene expression for protein-coding genes, where the nucleotide sequence of mRNA is decoded to synthesize proteins, the workhorses of cellular function. Understanding translation illuminates how genetic information ultimately produces the molecular machines, structural components, and regulatory molecules that determine cellular and organismal phenotypes. The genetic code defines how nucleotide triplets called codons specify amino acids. With 4 nucleotides, there are 64 possible 3-nucleotide combinations, but only 20 standard amino acids, making the code redundant or degenerate. Most amino acids are encoded by multiple codons. 3 codons, UAA, UAG, and UGA, are stop codons that signal translation termination. The codon AUG specifies methionine and also serves as the start codon initiating translation, meaning all proteins initially begin with methionine, though this is often removed post-translationally. The genetic code is nearly universal across all life, with only minor variations in mitochondria and some microorganisms. This universality indicates the code was established early in evolution and has been maintained across billions of years, constrained by the requirement that any code change would alter the meaning of many existing genes, likely causing catastrophic effects. The code structure shows non-random features. Chemically similar amino acids tend to be encoded by similar codons, meaning single nucleotide mutations often cause conservative amino acid substitutions rather than drastically different amino acids. Translation is carried out by ribosomes, large RNA protein complexes consisting of 2 subunits. The small subunit binds mRNA and matches codons with anticodons of transfer RNAs or tRNAs. The large subunit catalyzes peptide bond formation between amino acids. Bacterial ribosomes have sedimentation coefficients of 70s, with subunits of 30s and 50s. Eukaryotic cytoplasmic ribosomes are 80s with subunits of 40s and 60s. The larger eukaryotic ribosomes contain more proteins, but the catalytic core is structurally conserved, emphasizing RNA's central role. Transfer RNAs are adapter molecules connecting codons to amino acids. Each tRNA has an anticodon complementary to one or more codons and an acceptor stem where the corresponding amino acid attaches. Aminosyl tRNA synthetases catalyze attachment of amino acids to tRNAs, with one synthetase for each amino acid, ensuring correct pairing. The high fidelity of aminosylation, error rates below 1 in 10,000, is crucial for translation accuracy. Some synthetases have editing sites that hydrolyze incorrectly attached amino acids, increasing fidelity. Translation initiation in eukaryotes involves multiple steps and many initiation factors. The small ribosomal subunit, bound by initiation factors and carrying initiator methionine tRNA, binds the 5' cap of mRNA and scans for the start codon, typically the first AUG encountered. Scanning creates opportunities for regulation. Upstream open reading frames can interfere with translation of downstream coding sequences, and RNA secondary structures can inhibit scanning. Recognition of the start codon triggers initiation factor release and large subunit joining, forming a complete ribosome ready for elongation. Elongation proceeds through repetitive cycles, adding amino acids to the growing polypeptide chain. Each cycle involves 3 steps: binding of aminosyl tRNA to the ribosomal site in a codon-dependent manner, peptide bond formation transferring the growing chain to the new amino acid, and translocation moving the ribosome 3 nucleotides along mRNA, shifting tRNAs from A to P to E sites. Elongation factors facilitate these steps, with F2 delivering aminosyl tRNAs, EFG catalyzing translocation, and GTP hydrolysis providing energy and ensuring directionality. Peptide bonds form through nucleophilic attack of the amino group of the A site amino acid on the carbonyl carbon of the P site amino acid, transferring the growing chain to the A site. This peptidyl transferase reaction is catalyzed by ribosomal RNA, specifically the 23 srRNA in bacteria or the 28 srRNA in eukaryotes, making the ribosome a ribozyme. The discovery that RNA catalyzes peptide bond formation was surprising given the central importance of this reaction, and it supports the RNA world hypothesis that early life relied primarily on RNA catalysis. Translation elongates at approximately 4 to 20 amino acids per second in bacteria and approximately 3 to 8 amino acids per second in eukaryotes, much slower than DNA replication, but still rapid given the complexity of ensuring accuracy. Multiple ribosomes can translate single mRNAs simultaneously, forming polyribosomes or polysomes visible by electron microscopy. Polysome formation increases protein production from each mRNA, maximizing efficiency. Translation termination occurs when the ribosome encounters stop codons. No tRNAs recognize stop codons. Instead, release factors bind the site, promoting hydrolysis of the bond between the polypeptide and the P site tRNA, releasing the completed protein. The ribosome then dissociates into subunits, which can initiate translation on new mRNAs. In eukaryotes, Termination at stop codons triggers removal of the poly-A tail and degradation of the mRNA, coupling translation termination to mRNA decay. Translation fidelity relies on multiple discrimination steps. Initial codon-anticodon pairing provides specificity, but wobble-base pairing at the third codon position allows some tRNAs to recognize multiple codons. After initial binding, induced fit conformational changes in the ribosome occur only for correct codon-anticodon matches, creating a proofreading step. GTP hydrolysis following correct pairing is irreversible, ensuring that errors are not easily reversed. Overall error rates are approximately 1 incorrect amino acid per 10,000 incorporated, less accurate than DNA replication, but sufficient for protein function. Protein folding begins cotranslationally, with the emerging polypeptide folding as synthesis continues. Chaperone proteins bind nascent polypeptides, preventing aggregation and assisting folding. Some proteins require chaperone complexes, providing isolated chambers where folding occurs without interference from other cellular components. Proper folding is crucial for function, and misfolding can cause diseases including Alzheimer's, Parkinson's, and prion diseases. Cells have quality control systems that detect and degrade misfolded proteins. Post-translational modifications alter proteins after synthesis, expanding functional diversity beyond what primary sequence encodes. Modifications include phosphorylation regulating protein activity, methylation and acetylation affecting protein interactions, ubiquitination marking proteins for degradation, glycosylation adding sugar chains affecting protein stability and localization, and proteolytic cleavage activating or inactivating proteins. These modifications are often reversible, allowing dynamic regulation of protein function. Regulation of translation provides rapid control of protein levels without changing transcription. The 5' untranslated region of mRNAs contains regulatory elements including upstream open reading frames, internal ribosome entry sites, and sequences bound by RNA-binding proteins that modulate translation efficiency. Iron regulatory proteins bind iron-responsive elements in ferritin and transferrin receptor mRNAs, coordinating iron metabolism by reciprocally regulating these mRNAs. MicroRNAs bind 3' UTRs, repressing translation and promoting mRNA degradation. The signal recognition particle, or SRP pathway, directs proteins to the endoplasmic reticulum for secretion or membrane insertion. Signal sequences at protein N-termini are recognized by SRP, which pauses translation and directs the ribosome to ER membrane receptors. Translation resumes with the growing polypeptide threading through a translocan channel into the ER lumen or inserting into the membrane. This cotranslational translocation ensures that secreted and membrane proteins reach appropriate destinations without accumulating in the cytoplasm where they might misfold or aggregate. Mutations are changes in DNA sequence that can arise through errors, damage, or recombination. Mutations represent permanent changes in DNA sequence, providing the raw material for evolution while also causing diseases and genetic disorders. Understanding mutation mechanisms, types, and consequences illuminates both evolutionary processes and medical genetics while revealing the balance between genome stability and variation. Mutations are classified by their scale and mechanism. Point mutations affect single nucleotides, including substitutions where one nucleotide replaces another, and insertions or deletions of single nucleotides. Larger-scale mutations include insertions and deletions of multiple nucleotides, inversions where sequence orientation reverses, duplications creating additional copies of sequences, and translocations moving sequences between chromosomes. Chromosomal abnormalities involve changes to chromosome number or structure visible cytogenetically. Substitution mutations are divided into transitions, purine to purine or pyrimidine to pyrimidine changes, and transversions, purine to pyrimidine or vice versa changes. Transitions are more common than transversions, occurring approximately twice as frequently, possibly because they require smaller structural changes. The effects of substitutions depend on their locations and the nature of the change. Synonymous or silent mutations don't change the amino acid due to genetic code degeneracy. Missense mutations change the amino acid, with effects depending on whether the change is conservative or non-conservative. Nonsense mutations create stop codons, usually truncating proteins. Insertions and deletions, collectively called indels, have different effects depending on their size and location. Encoding sequences, indels that are not multiples of 3 nucleotides, cause frameshift mutations, shifting the reading frame for all downstream codons. Frameshifts typically create non-functional proteins because downstream sequence is translated incorrectly and premature stop codons usually occur. Indels that are multiples of 3 add or remove amino acids without frameshifting, potentially preserving some protein function. Spontaneous mutations arise without external mutagenic agents through inherent limitations of cellular processes. DNA replication errors occur despite polymerase proofreading and mismatch repair, with error rates after all error correction approximately 1 mistake per billion nucleotides replicated. Spontaneous DNA damage occurs continuously from water, reactive oxygen species, and spontaneous chemical changes. Deamination of cytosine produces uracil, which if unrepaired pairs with adenine rather than guanine during replication, causing C to T transitions. Depurination, loss of purine bases, creates a basic sites that can lead to mutations if not repaired. Induced mutations result from exposure to mutagens, agents that damage DNA or interfere with replication. Physical mutagens include ultraviolet radiation causing thymine dimers, ionizing radiation producing double-strand breaks and base modifications, and heat accelerating spontaneous damage. Chemical mutagens include base analogues incorporated during replication, intercalating agents inserting between bases causing frameshift mutations, alkylating agents adding alkyl groups to bases altering pairing, and oxidizing agents damaging bases. Biological mutagens include transposable elements inserting into genes and viruses integrating into genomes. The mutation rate, the probability of mutation per nucleotide per generation, varies across genomes and organisms. In humans, the germline mutation rate averages approximately 1.2 times 10 to the -8 per base pair per generation, meaning each person carries approximately 60 to 70 new mutations not present in either parent. Mutation rates are higher in males than females and increase with paternal age, reflecting more cell divisions in spermatogenesis and accumulation of errors over time. Mutation rates vary along chromosomes, with higher rates near sequences prone to errors or in regions with reduced repair efficiency. Hotspots are sequences with elevated mutation rates due to sequence properties or local conditions. CpG dinucleotides, cytosine followed by guanine, are hotspots because cytosines in CpG contexts are often methylated, and methylated cytosines frequently deaminate to thymine, causing C to T transitions. Repetitive sequences, including microsatellites and homopolymer runs, have elevated mutation rates because polymerases frequently slip during replication of repeated sequences, causing insertions or deletions. Recombination hotspots have elevated mutation rates because recombination processes can introduce errors. Selection determines mutation fate in populations. Deleterious mutations reducing fitness are eliminated by purifying selection, though recessive mutations can persist in heterozygotes. Beneficial mutations increasing fitness spread through positive selection, though initial frequency is low, making them vulnerable to loss by drift. Neutral mutations, those not affecting fitness, drift randomly, with some eventually becoming fixed by chance. Most mutations are neutral or deleterious. Beneficial mutations are rare, but they provide the substrate for adaptation and evolution. The distribution of fitness effects, the spectrum of selective effects for new mutations, is skewed toward deleterious effects. Most mutations that change protein sequences are deleterious because proteins are finely tuned by evolution, and random changes usually reduce function. Estimates suggest approximately 70% of amino acid changing mutations are deleterious, approximately 25% are neutral, and approximately 5% are beneficial. For mutations in regulatory sequences, effects are harder to predict, but similar principles apply. Most changes to optimized regulatory networks reduce fitness. Germline mutations occur in reproductive cells and are transmitted to offspring, contributing to evolution and hereditary disease. Somatic mutations occur in non-reproductive cells and are not inherited, though they can cause problems in the individual, including cancer. The distinction is important. Germline mutations affect descendants and contribute to species evolution, while somatic mutations affect only the individual. However, in organisms with vegetative reproduction, the distinction blurs because somatic lineages can become germline. Cancer illustrates somatic mutation consequences. Most cancers accumulate multiple driver mutations, mutations conferring selective advantage for cancer cells, in oncogenes promoting growth or tumor suppressors restraining growth. Additional passenger mutations, mutations not contributing to cancer but occurring in the lineage, accumulate as cancer evolves. The multi-hit hypothesis proposes that cancer requires accumulation of multiple mutations, disabling multiple safeguards, explaining why cancer incidence increases with age as mutations accumulate. DNA repair systems maintain genome integrity by detecting and correcting damage before it becomes mutation. Base excision repair removes damaged bases through glycosylases recognizing specific lesions. Nucleotide excision repair removes bulky lesions, including thymine dimers, through recognition, excision, and resynthesis. Mismatch repair corrects replication errors by recognizing mismatches, excising the newly synthesized strand, and resynthesizing correctly. Double-strand break repair occurs through homologous recombination or non-homologous end joining. Defects in repair systems cause increased mutation rates and cancer susceptibility. Transposable elements or transposons are DNA sequences that can move within genomes, creating mutations by inserting into genes or regulatory sequences. Class I transposons or retrotransposons move through RNA intermediates transcribed from genomic copies, reverse transcribed, and inserted elsewhere. Class II transposons or DNA transposons move directly through DNA intermediates. Transposons comprise approximately 45% of the human genome, though most are inactive relics. Active transposons can cause genetic instability and disease, but they also contribute to genome evolution and occasionally provide regulatory sequences or create useful mutations. The relationship between genotype and phenotype is complex and nonlinear. Many mutations have no phenotypic effect, occurring in non-functional sequences or causing silent changes. Some mutations have subtle effects detectable only under specific conditions. Others have major effects, causing disease or lethality. Epistasis, interaction between genes, means mutation effects depend on genetic background. Pleiotropy, single genes affecting multiple traits, means mutations can have diverse effects. Understanding these complexities is essential for predicting mutation consequences and interpreting genetic variation. Inheritance patterns determine how genetic variation passes from parents to offspring. The principles of inheritance were established by Gregor Mendel in the 19th century, before DNA was identified as the genetic material, but they remain foundational for understanding genetics. Modern molecular knowledge provides mechanistic understanding of Mendelian inheritance while also revealing complexities, including linkage, epigenetics, and non-Mendelian patterns. Mendel's first law, or the law of segregation, states that each organism carries two copies of each gene, called alleles, that segregate during gamete formation so that each gamete receives one copy. This law reflects the diploid nature of most organisms, where chromosomes occur in pairs, one inherited from each parent. During meiosis, homologous chromosomes separate, ensuring that gametes are haploid with single chromosome sets. Fertilization restores diploidy by combining gametes from two parents. Mendel's second law, or the law of independent assortment, states that genes for different traits segregate independently during gamete formation. This law holds for genes on different chromosomes, which segregate independently during meiosis because chromosomes assort randomly. However, genes on the same chromosome violate independent assortment because they tend to be inherited together. They're genetically linked. The frequency of recombination between linked genes reflects their physical distance along chromosomes, providing the basis for genetic mapping. Dominant and recessive alleles show different inheritance patterns. Dominant alleles manifest their effects in heterozygotes, individuals carrying one dominant and one recessive allele, while recessive alleles only manifest in homozygotes, individuals carrying two recessive alleles. Dominance reflects molecular mechanisms. Dominant alleles often produce functional proteins at levels sufficient for normal phenotype, even when only one copy is present, while recessive alleles typically represent loss of function, requiring both copies to be defective for phenotype to appear. Punnett squares predict offspring genotype and phenotype frequencies for simple crosses. For a cross between heterozygotes AA times AA, the Punnett square shows 1/4 AA, 1/2 AA, and 1/4 AA genotypic frequencies, and 3/4 dominant to 1/4 recessive phenotypic frequencies. If it is completely dominant over A, these ratios match Mendel's observations and reflect the random segregation of alleles during gamete formation and random combination during fertilization. Incomplete dominance occurs when heterozygotes show intermediate phenotypes between homozygotes. For example, red and white flower alleles might produce pink flowers in heterozygotes. Codominance occurs when both alleles manifest in heterozygotes. The Abo blood group shows codominance, with AB individuals expressing both A and B antigens. These patterns reveal that dominance is not an intrinsic property of alleles, but rather reflects how gene products interact to produce phenotypes. Sex-linked inheritance involves genes on sex chromosomes, particularly X-linked genes showing distinctive patterns. In mammals, including humans, females have 2 X chromosomes while males have 1 X, and 1 Y. X-linked recessive traits like color blindness and hemophilia affect males more frequently than females, because males need only 1 mutant allele to express the phenotype, while females need 2. Females heterozygous for X-linked traits are carriers who don't express the trait, but can transmit it to offspring. X inactivation in female mammals randomly silences 1 X chromosome in each cell during early development, equalizing X-linked gene expression between males with 1 X and females with 2. The choice of which X to inactivate is random, creating mosaic patterns where some cells express the maternal X, while others express the paternal X. Calico cats exemplify X inactivation mosaicism. The orange and black patches reflect random inactivation of X chromosomes carrying different color alleles in different cell lineages. Y-linked or Hollandric inheritance involves genes on the Y chromosome transmitted exclusively from fathers to sons. The human Y chromosome is small with few genes, most involved in male sex determination and spermatogenesis. Y-linked traits are rare in medical genetics, but important for evolutionary studies, because Y chromosomes, like mitochondrial DNA, don't recombine in most of their length, preserving haplotypes that track paternal lineages through populations. Mitochondrial inheritance follows maternal patterns because eggs contribute nearly all cytoplasm to zygotes, while sperm contribute primarily nuclei. Mitochondrial DNA encodes 37 genes, all maternally inherited. Mitochondrial diseases result from mutations in mitochondrial DNA, showing distinctive inheritance patterns where affected mothers transmit disease to all children, but only daughters transmit to subsequent generations. Heteroplasmy, cells containing mixtures of mutant and normal mitochondria, creates variable expression depending on the proportion of mutant mitochondria. Genomic imprinting represents parent of origin effects where gene expression depends on whether alleles were inherited from mothers or fathers. Imprinted genes show monoallelic expression, with 1 allele silenced by epigenetic modifications. Approximately 150 human genes are imprinted, many involved in growth regulation. Imprinting disorders including Prader-Willi and Angelman syndromes result from deletions or abnormal methylation of imprinted regions, with phenotypes depending on whether maternal or paternal alleles are affected. Multifactorial or polygenic inheritance involves multiple genes contributing to phenotypes, often with environmental influences. Height, skin color, intelligence, and disease susceptibility exemplify multifactorial traits showing continuous variation rather than discrete categories. Each contributing gene has small effects, but collectively they produce the phenotypic range observed. Heritability, the proportion of phenotypic variation due to genetic variation, quantifies genetic contributions for multifactorial traits, though heritability is population-specific and context-dependent. Linkage disequilibrium describes non-random associations between alleles at different loci, reflecting physical linkage, population history, or selection. Linked genes tend to be inherited together more often than expected by chance, violating independent assortment. Recombination during meiosis breaks down linkage disequilibrium over generations, with the rate depending on physical distance. Closer genes show stronger linkage. Linkage analysis maps disease genes by identifying markers that co-segregate with disease in families. Quantitative trait loci, or QTL, are chromosomal regions containing genes affecting quantitative traits. QTL mapping identifies genomic regions associated with phenotypic variation through statistical analysis of genotype-phenotype correlations in segregating populations. Modern genome-wide association studies, or GWAS, extend QTL mapping to humans, testing associations between millions of genetic variants and traits in large populations. GWAS have identified thousands of variants influencing complex diseases, though individual effects are often small, and most variants are in non-coding regions, complicating functional interpretation. Pedigree analysis traces trait inheritance through families, revealing inheritance patterns and predicting recurrence risks. Symbols represent individuals, relationships, and phenotypes, allowing visualization of inheritance across generations. Pedigrees distinguish autosomal dominant, autosomal recessive, X-linked, and mitochondrial inheritance patterns. Genetic counselors use pedigrees to assess disease risk and inform reproductive decisions. Molecular testing increasingly complements pedigree analysis, directly identifying causative mutations. Genetic engineering techniques allow precise manipulation of DNA sequences in living organisms. The ability to deliberately modify DNA sequences has revolutionized biology, medicine, and agriculture, transforming genetics from an observational science to an experimental one capable of testing hypotheses through designed genetic changes. Understanding genetic engineering techniques provides insights into their applications and limitations, while raising important ethical considerations. Restriction enzymes or restriction endonucleases are bacterial proteins that cut DNA at specific recognition sequences, typically 4 to 8 base pairs long. These enzymes evolved as bacterial immune systems against viruses, but molecular biologists adapted them as tools for cutting DNA at defined positions. Different restriction enzymes recognize different sequences, creating a toolkit for cutting DNA predictably. Some create blunt ends where both strands are cut at the same position, while others create sticky or cohesive ends with single-stranded overhangs. DNA ligase joins DNA fragments by catalyzing phosphodiester bond formation between adjacent nucleotides, sealing nicks in the sugar-phosphate backbone. Ligase can join blunt ends, though this is inefficient, or join complementary sticky ends, which is much more efficient because base pairing holds fragments together while ligase acts. The combination of restriction enzymes to cut DNA at specific sites and ligase to join fragments enables the creation of recombinant DNA molecules combining sequences from different sources. Cloning involves inserting DNA fragments into vectors, DNA molecules that can replicate autonomously, and propagating them in host cells. Plasmids are circular DNA molecules from bacteria that replicate independently of chromosomal DNA, making them ideal vectors. To clone a gene, the gene is cut from genomic DNA using restriction enzymes. inserted into a plasmid cut with the same enzyme, ligated, and introduced into bacteria by transformation. Bacteria carrying recombinant plasmids are selected, grown in culture, and used to produce large quantities of the cloned DNA. The polymerase chain reaction, or PCR, amplifies specific DNA sequences exponentially through repeated cycles of denaturation, annealing, and extension. Each cycle doubles the target sequence, so 30 cycles produce over 1 billion copies. PCR requires primers, short DNA sequences complementary to the target sequence ends, and heat-stable DNA polymerase, such as Taq polymerase from thermophilic bacteria. PCR revolutionized molecular biology by enabling amplification of DNA from minimal starting material, facilitating forensics, diagnostics, research, and numerous applications. Gel electrophoresis separates DNA fragments by size through application of electric current to DNA samples in agarose or polyacrylamide gels. DNA is negatively charged due to phosphate groups, so it migrates toward the positive electrode when current is applied. Smaller fragments migrate faster through the gel matrix than larger fragments, creating separation by size. After electrophoresis, DNA is visualized using fluorescent dyes that bind DNA, revealing banding patterns that can be analyzed to determine fragment sizes or identify specific sequences. DNA sequencing determines the nucleotide sequence of DNA molecules. Sanger sequencing, developed in the 1970s, uses dideoxynucleotides, modified nucleotides lacking the 3' hydroxyl group needed for chain extension, to terminate synthesis at specific positions. By incorporating fluorescently labeled dideoxynucleotides for each of the 4 bases and separating fragments by size, the sequence is read from the pattern of terminations. Modern capillary electrophoresis automates Sanger sequencing, achieving high throughput though limited compared to next-generation methods. Next-generation sequencing, or NGS technologies, including Illumina sequencing, PacBio sequencing, and Oxford nanopore sequencing, have revolutionized genomics by massively parallelizing sequencing reactions, generating billions of sequence reads simultaneously. Illumina sequencing, the most widely used NGS method, sequences millions of DNA fragments simultaneously on flow cells through cycles of reversible termination, achieving hundreds of gigabases of sequence per run. These technologies enabled whole genome sequencing to become routine, with human genomes now sequenced for under $1,000. CRISPR-Cas9 represents the most powerful genetic engineering tool developed to date, enabling precise editing of genes in living cells and organisms. CRISPR, clustered regularly interspaced short palindromic repeats, are sequences in bacterial genomes that store records of past viral infections. Cas9 is an endonuclease that uses RNA guides to target and cut specific DNA sequences. The system was adapted for genome editing by providing synthetic guide RNAs directing Cas9 to desired targets, creating double-strand breaks that cells repair, often incorporating designed changes. CRISPR editing can create knockouts, genes inactivated by frameshift mutations introduced during error-prone repair of breaks, or knock-ins, specific sequences inserted at breaks when provided as repair templates. The ease of designing guide RNAs and the high efficiency of editing have made CRISPR accessible to labs worldwide, accelerating research across biology and medicine. CRISPR has been used to create disease models, screen gene functions, develop therapies, engineer crops and modify organisms in ways previously impossible or impractical. Gene therapy aims to treat genetic diseases by introducing functional genes to replace defective ones or modifying disease-causing genes. Viral vectors, including adeno-associated viruses or AAVs, are commonly used to deliver genes to cells, taking advantage of viral machinery for entering cells and expressing genes. AAV vectors have been used successfully to treat inherited blindness and other diseases. CRISPR-based therapies now in clinical trials aim to correct mutations directly rather than adding supplementary genes, potentially providing permanent cures. Transgenic organisms carry foreign genes integrated into their genomes. Transgenic mice are created by injecting DNA into fertilized eggs before implantation, allowing the study of gene function and creation of disease models. Transgenic crops, including herbicide-resistant and insect-resistant varieties, have been widely adopted in agriculture. Genetically modified animals produce pharmaceuticals in milk or organs for xenotransplantation. The creation of transgenic organisms raises ethical and ecological concerns requiring careful consideration. Gene drives are genetic engineering systems that bias inheritance of engineered alleles, spreading them through populations even if they reduce fitness. CRISPR-based gene drives work by copying the engineered allele to the homologous chromosome during DNA repair, converting heterozygotes to homozygotes. Gene drives have been proposed for controlling disease vectors like mosquitoes or invasive species, but their potential to spread uncontrollably through populations raises significant concerns, requiring extensive safety research and regulatory frameworks before environmental release. Synthetic biology combines engineering principles with genetic engineering to design and build biological systems with novel functions. Synthetic biologists design genetic circuits controlling cellular behaviors, engineer metabolic pathways producing valuable compounds, and even design minimal genomes defining the essential genes for life. The field aims to make biology more predictable and programmable, enabling applications from biosensors detecting pollutants to microbes producing biofuels. Ethical questions about creating new life forms motivate ongoing dialogue. The Human Genome Project revealed the complete DNA sequence of human chromosomes. The Human Genome Project represents one of biology's most ambitious undertakings, determining the complete nucleotide sequence of human DNA and cataloging all human genes. Completed in 2003, after 13 years of international collaboration, the project revolutionized biology and medicine while raising questions about genomics implications for society. The project officially began in 1990 with the goal of sequencing the approximately 3 billion base pairs comprising the human genome. Initial estimates suggested the project would take 15 years and cost $3 billion, approximately $1 per base pair. The project involved 20 centers in 6 countries, with the United States contributing the largest share through the National Institutes of Health and Department of Energy. International collaboration and data sharing were fundamental principles, with all sequenced data released publicly without restrictions. The sequencing strategy involved creating libraries of DNA fragments from human genomes, sequencing individual fragments, and assembling fragments into continuous sequences using overlaps. 2 main approaches were used: hierarchical shotgun sequencing, which first mapped fragments to chromosomes before sequencing, and whole genome shotgun sequencing, which sequenced random fragments and used computational assembly. Both approaches contributed to the final sequence, with the public project primarily using hierarchical methods, while Celera Genomics, a private company, used whole genome shotgun sequencing. The draft sequence was announced in June 2000 with approximately 90% coverage, and the complete sequence was finished in April 2003, 50 years after Watson and Crick's double helix structure determination. The finished sequence had error rates below 1 error per 100,000 bases and gaps totaling less than 1%, representing extraordinary accuracy and completeness. The achievement demonstrated the power of large-scale scientific collaboration and technology development. The human genome contains approximately 3.2 billion base pairs distributed across 23 chromosome pairs, 22 autosomes plus X and Y sex chromosomes. Chromosome 1 is the largest with approximately 250 million base pairs, while chromosome 21 is the smallest autosome with approximately 50 million base pairs. The Y chromosome contains approximately 59 million base pairs, but much is repetitive, and only the male-specific region of approximately 25 million base pairs was fully sequenced initially. Perhaps the most surprising finding was the small number of protein-coding genes, approximately 20,000 to 25,000, far fewer than pre-project estimates of up to 100,000 genes. This revealed that organismal complexity doesn't correlate simply with gene number, and that humans achieve complexity through alternative splicing, gene regulation, and other mechanisms, rather than having vastly more genes than simpler organisms. The finding humbled assumptions about human genetic exceptionalism. Protein-coding sequences comprise only approximately 1.5% of the genome, raising questions about what the remaining 98.5% does. Much consists of repetitive sequences, including transposable elements, accounting for approximately 45% of the genome. Introns, regulatory sequences, and intergenic regions account for much of the remainder. The encode project later found that approximately 80% of the genome is transcribed, though the functional significance of this pervasive transcription remains debated. The genome contains approximately 3 million single nucleotide polymorphisms, or SNPs, positions where individuals differ by single nucleotides, with each person's genome differing from the reference sequence at approximately 4 to 5 million positions. These variants provide the genetic diversity underlying phenotypic differences among individuals and populations. Most variants are in non-coding regions and have no functional effects, but variants in coding or regulatory sequences can affect traits and disease susceptibility. Genome structure revealed that genes are not uniformly distributed but cluster in GC-rich regions while being sparse in AT-rich regions. GC content varies across the genome from approximately 30% to 60% in different regions, creating isochores, large regions with relatively uniform GC content. The reasons for this heterogeneity relate to recombination rates, mutation patterns, and selection, though details remain incompletely understood. Gene density varies from 0 genes per megabase in some regions to over 20 genes per megabase in others. Segmental duplications, sequences longer than 1 kilobase with over 90% sequence identity, comprise approximately 5% of the genome. These duplications, resulting from evolutionary events copying chromosomal segments, create regions of high sequence similarity that complicate sequence assembly, and can cause problems during meiosis when misalignment leads to deletions or duplications. Segmental duplications have contributed to evolutionary innovation by providing raw material for the evolution of new genes. CpG islands are regions enriched for CG dinucleotides, often associated with gene promoters. Despite CG dinucleotides being generally underrepresented in the genome due to mutation of methylated cytosines to thymines over evolutionary time, CpG islands maintain high CG content, possibly because they're protected from methylation. Identifying CpG islands helps locate genes and regulatory regions, making them useful markers for genome annotation. The reference genome sequence provides a foundation, but doesn't capture all human genetic diversity. Subsequent projects, including the 1000 Genomes Project, sequenced genomes from thousands of individuals worldwide, cataloging genetic variation across populations. These efforts revealed that most variants are rare. Each person carries many variants found in few others, and that different populations have different variant frequencies, reflecting demographic history and local adaptation. Understanding population variation is crucial for interpreting genetic associations and developing precision medicine. Applications of genome sequencing have proliferated since the Human Genome Project. Clinical sequencing identifies disease-causing mutations, guiding diagnosis and treatment. Cancer genomics reveals somatic mutations driving tumor growth, informing targeted therapies. Pharmacogenomics uses genetic variants to predict drug responses, enabling personalized medication selection. Prenatal and newborn screening detect genetic disorders early when interventions can help. Direct-to-consumer genetic testing provides ancestry information and health risk assessments, though with limitations regarding interpretation and accuracy. The decreasing cost of sequencing, from billions of dollars for the first genome to hundreds of dollars today, has made sequencing routine in research and increasingly common in healthcare. Whole genome sequencing, whole exome sequencing targeting just coding regions, and targeted gene panels for specific conditions offer different cost accuracy trade-offs. The interpretation of variants remains challenging. Distinguishing pathogenic variants from benign ones requires sophisticated analysis integrating evolutionary conservation, functional predictions, and clinical observations. Genomics and bioinformatics analyze genome-scale data to understand biological systems. The massive datasets generated by genome sequencing and related technologies require computational approaches for storage, analysis, and interpretation. Genomics, the study of entire genomes, and bioinformatics, the development and application of computational tools for biological data, have become central to modern biology, transforming how research is conducted and how biological questions are addressed. Sequence alignment is a fundamental bioinformatics task involving arranging sequences to identify regions of similarity, indicating functional, structural, or evolutionary relationships. Pairwise alignment compares two sequences, while multiple sequence alignment compares three or more, revealing conserved regions shared across sequences. Alignment algorithms including BLAST, Basic Local Alignment Search Tool, efficiently search databases containing billions of sequences to find matches, enabling identification of genes, prediction of function, and evolutionary analysis. Genome annotation is the process of identifying features in genome sequences, including genes, regulatory elements, and repetitive sequences. Gene prediction algorithms identify coding sequences through signals including start and stop codons, splice sites, and open reading frames, though accuracy is imperfect, particularly for complex eukaryotic genes with multiple small exons. Experimental data, including RNA sequencing, improves annotation by providing direct evidence of transcription. Functional annotation assigns biological functions to genes through sequence similarity to characterize genes, though many genes remain functionally uncharacterized. Comparative genomics compares genomes across species to identify conserved and lineage-specific features, infer evolutionary relationships, and predict function. Sequences conserved across distant species are likely functionally important. Purifying selection prevents their divergence, while lineage-specific sequences may contribute to unique adaptations. The comparison of human and chimpanzee genomes, which differ by approximately 1.2% in aligned sequences, has identified human-specific changes, including accelerated evolution of genes involved in brain development. Phylogenetic analysis reconstructs evolutionary relationships through comparisons of DNA or protein sequences. Phylogenetic trees depict hypothesized relationships with branch lengths representing evolutionary distances. Methods including maximum parsimony, maximum likelihood, and Bayesian inference use different criteria to identify optimal trees. Molecular phylogenetics has revolutionized understanding of life's tree, revealing relationships invisible from morphology alone and resolving controversies about evolutionary history. Transcriptomic studies RNA populations in cells through RNA sequencing or RNA-seq, which sequences all RNAs, providing genome-wide expression profiles. RNA-seq reveals which genes are active, at what levels, with what splice variants, and how expression changes across conditions, tissues, or developmental stages. Differential expression analysis identifies genes with significantly different expression between conditions, suggesting involvement in biological processes of interest. Single-cell RNA-seq profiles individual cells, revealing cellular heterogeneity invisible in bulk samples. Functional genomics aims to understand gene functions and interactions at genome scale. Approaches include systematic gene knockout or knockdown screens testing effects of inactivating each gene, overexpression screens testing effects of increasing gene expression, protein-protein interaction mapping identifying physical associations, and genome-wide association studies linking genetic variants to phenotypes. These approaches generate massive datasets requiring computational analysis to identify patterns and generate hypotheses. Systems biology integrates genomic, transcriptomic, proteomic, and metabolomic data to understand biological systems holistically rather than focusing on individual genes or proteins. Network analysis represents biological systems as networks of interacting components, genes, proteins, metabolites, and analyzes network properties to understand system behavior. Mathematical modeling simulates system dynamics, testing hypotheses about regulatory mechanisms and predicting system responses to perturbations. Systems approaches are particularly valuable for understanding complex traits and diseases involving many genes and environmental factors. Metagenomic sequences DNA from environmental samples containing mixed populations of organisms, providing insights into microbial communities without requiring cultivation. Human microbiome studies using metagenomics have revealed that each person harbors trillions of bacterial cells representing thousands of species, with communities varying across body sites and influencing health and disease. Environmental metagenomics studies ocean, soil, and other ecosystems, discovering enormous microbial diversity, including many novel lineages. Epigenomics maps epigenetic modifications across genomes, including DNA methylation, histone modifications, and chromatin accessibility. Bisulfite sequencing converts unmethylated cytosines to uracils while leaving methylated cytosines unchanged, allowing determination of methylation patterns. ChIP-seq maps histone modifications by using antibodies to immunoprecipitate modified histones along with associated DNA, then sequencing the DNA. ATAC-seq maps chromatin accessibility, revealing open chromatin regions likely to be regulatory elements. These approaches reveal regulatory landscapes controlling gene expression. Structural variation analysis identifies large-scale genomic differences including deletions, duplications, inversions, and translocations. While SNPs affect single nucleotides, structural variants affect larger segments from thousands to millions of base pairs. Structural variants contribute substantially to genetic diversity and disease, affecting gene dosage, disrupting genes, or altering regulatory elements. Detecting structural variants is challenging, requiring specialized algorithms analyzing read depth, discordant read pairs, or split reads, or using long-read sequencing technologies that span entire variants. Genome-wide association studies, or GWAS, test associations between genetic variants and traits in large populations, typically testing hundreds of thousands to millions of SNPs. GWAS have identified thousands of variants associated with complex diseases including diabetes, heart disease, schizophrenia, and Alzheimer's disease, and with traits including height, body mass index, and educational attainment. However, most associated variants have small effects and are in non-coding regions, complicating interpretation and limiting predictive ability. Polygenic risk scores combining effects of many variants show promise for disease prediction. Machine learning applies computational algorithms that improve through experience to biological problems, including predicting gene function, classifying disease subtypes, interpreting genomic variants, and analyzing images. Deep learning using multi-layered neural networks has achieved remarkable success in tasks including protein structure prediction, variant effect prediction, and gene expression modeling. As biological datasets grow, machine learning will become increasingly important for extracting insights from complex, high-dimensional data. If you're still awake somewhere in this exploration of DNA, of double helices and base pairs, of replication and transcription, of genes and genomes, of mutations and inheritance, there's more to discover about the molecule that encodes life's instructions. But if sleep has found you, if consciousness has faded into dreams, then rest knowing that in every cell of your body, DNA continues its patient work of storing, expressing, and transmitting the information that makes you who you are. While you sleep tonight, DNA in your cells undergoes continuous maintenance and repair. Damage from normal metabolism, background radiation, and spontaneous chemical reactions affects thousands of bases daily, yet repair systems work constantly to detect and correct this damage. Mismatch repair proteins scan newly replicated DNA for errors. Base excision repair removes damaged bases. Nucleotide excision repair cuts out bulky lesions. The fidelity of these systems maintains genome integrity through decades of life, though some damage escapes repair, accumulating as you age. Your cells aren't dividing while you sleep. Most are in interphase, the growth phase between divisions, but in some tissues, cells are preparing for division. In bone marrow, stem cells are dividing to produce new blood cells, replacing those that have aged. In intestinal crypts, epithelial stem cells divide to replace the lining of your digestive tract, which renews completely every few days. In these dividing cells, DNA replication is occurring, with replisomes moving along chromosomes, synthesizing new strands with remarkable accuracy, ensuring that daughter cells receive complete, accurate genome copies. The genes active in your cells right now reflect your cell types and current physiological state. In liver cells, genes for metabolic enzymes are highly expressed, processing nutrients and detoxifying compounds. In neurons, genes for neurotransmitter synthesis and synaptic proteins are active, maintaining neural function. In muscle cells, genes for contractile proteins are expressed, though muscles are relaxed during sleep. The circadian clock genes in all cells are following their approximately 24-hour cycles, with some genes expressed now that won't be expressed during daytime. The molecular machinery transcribing and translating genes operates continuously. RNA polymerases are transcribing thousands of genes simultaneously across your genome, producing mRNAs that will be translated into proteins. Ribosomes are translating these mRNAs, adding amino acids to growing polypeptide chains that will fold into functional proteins. Chaperones are assisting protein folding, ensuring correct three-dimensional structures. Protein degradation systems are breaking down damaged or unneeded proteins, maintaining protein quality and cellular homeostasis. The information in your DNA, your genome, is uniquely yours, differing from every other person's genome by millions of variants. Some variants you inherited from your mother, others from your father, creating your unique combination. A few variants, approximately 60 to 70, arose as new mutations in your parents' germ cells or during your early development, making them unique to you. These variants influence your traits, appearance, physiology, behavior, though in complex ways involving interactions among genes and with environment. Your genome also contains history, evolutionary history spanning billions of years. The genes you carry are descended from genes in ancient organisms, modified through mutation and selection over countless generations. Some genes are nearly identical to those in distantly related species, reflecting their essential functions maintained by purifying selection. Others have diverged, acquiring new functions or specializations. Transposable elements filling much of your genome are relics of ancient infections, now mostly domesticated though occasionally causing mutations. If you have children, you'll transmit approximately half your genome to each child through your germ cells, eggs if you're female, sperm if you're male. Meiosis, the specialized cell division producing germ cells, shuffles your maternal and paternal chromosomes randomly, creating millions of possible combinations. Recombination between homologous chromosomes during meiosis creates even more variation, combining segments from maternal and paternal chromosomes into new configurations. Each child receives a unique subset of your genetic variation, combined with a unique subset from their other parent. The mutations accumulating in your cells throughout life won't be transmitted to children. Only germline mutations in reproductive cells are heritable, but somatic mutations in your body's cells accumulate with age, creating mosaicism where different cells carry different mutations. Most somatic mutations have no effect, but some contribute to aging, disease, or cancer. The mutation rate in somatic cells is similar to germline rates, approximately 1 mutation per cell division, meaning trillions of mutations occur across your body's trillions of cells over your lifetime. Understanding DNA has transformed medicine, enabling diagnosis of genetic diseases, prediction of disease risk, development of targeted therapies, and personalized treatment based on individual genotypes. The DNA in your cells could be sequenced tomorrow, revealing variants affecting your health risks, drug responses, and ancestry. This information has power to inform healthcare decisions while also raising privacy concerns and questions about genetic determinism versus the importance of environment and choices. The same DNA molecule stores hereditary information across all life, from bacteria to plants to animals, using the same genetic code and mechanisms. This universality reflects life's common origin, all organisms descending from ancient ancestors that established DNA as the genetic material billions of years ago. The conservation of DNA structure and function across life enables genetic engineering, allowing genes to be transferred between organisms, creating insulin-producing bacteria, pest-resistant crops, and gene therapies for human diseases. The future of DNA science promises continued advances. Improved sequencing technologies will make genome sequencing faster and cheaper. Better understanding of genome organization and regulation will reveal how genes are controlled. Advances in genetic engineering will enable more precise modifications for research and therapy. Synthetic biology will create organisms with designed genomes, but fundamental questions remain. How does genome sequence encode organismal form? How do genotype and environment interact to produce phenotype? How can we predict mutation effects and disease risk? Consider, as you drift towards sleep or have already fallen, the remarkable molecule in every cell of your body. A molecule only 2 nanometers wide, but meters long when stretched. A molecule storing 3 billion bits of information in chemical form. A molecule that can be copied with extraordinary fidelity, transmitting information across generations. A molecule that can change through mutation, creating variation on which natural selection acts. A molecule that connects you to all life through common origin and shared mechanisms. Your DNA is both ancient and unique. Ancient in that it carries sequences conserved across millions of years of evolution. Unique in that your particular combination of variants exists nowhere else. It's both stable and dynamic, stable enough to preserve information through cell divisions and generations, dynamic enough to evolve and adapt. It's both simple and complex, simple in its chemical structure of 4 bases paired by hydrogen bonds, complex in how it encodes the instructions for building and maintaining organisms. See you next time.